Okay. Hi, everybody. I hope that everybody can hear me. So that's Steph from Ongava Research Center, and thanks for joining us today. Today we have a seminar from Jessy Patterson with one of our PhD students in collaboration with UGA. And Jim Beasley, she's going to talk to us about work that she's been doing on Ongava Game Reserve. And globally, her PhD is on the effect of anthropogenic activities on mammals' behavior. And today she's talking about activity patterns in actually behavior at waterholes on Ongava. Over to you, Jessy.
Okay. Thanks, Steph. So, yep, my name's Jessy. Hold on. There we go. My name's Jessy Patterson. I'm a PhD candidate at the University of Georgia, working with Dr. Jim Beasley, and we collaborated with Noma and Steph here at the Ongava Research Center to look at the effects of human presence on mammal activity patterns and waterhole attendance. Sorry, technical difficulties. Okay, so we all know that human impacts are increasing both on the environment and wildlife populations across the globe. And carnivores and ungulates are of particular concern because they have declined substantially across the world over the last 40 to 50 years. And since they play really important roles in both food web dynamics and ecosystem processes, it's important that we work to fully understand how any of our conservation efforts might be helping to preserve ecosystem health and biodiversity across the world as well. We also know that wildlife are exposed to human presence in many ways, one of these being through wildlife-based tourism. And while tourism can be really beneficial for increasing conservation efforts or educating tourists or providing employment, it's been documented to increase fear, aggression, and vigilance in some mammals. And there's a lot that we don't really understand in terms of the effects of tourism and human presence on wildlife populations. Another way that wildlife are often exposed to human presence is through wildlife surveys, specifically through game counts that are conducted for conservation efforts. And consistent monitoring is really important in protected areas where biodiversity and tourism contribute to conservation goals, but it's important that we understand how that human presence may be biasing these population estimates or altering animal activity patterns, which can have cascading effects throughout the ecosystem. So for this study, we were focusing on trying to determine if human presence or the presence of permanent infrastructures that are used by humans can affect mammal visits or their temporal activity patterns at waterholes. The study took place in September of 2016 and 2022 here at the Ongava Game Reserve in Namibia. And we used 12 waterholes for this study. So I won't go into detail about where the Ongava Game Reserve is located, but from this map here, you can see that the 12 waterholes are fairly equally distributed across the landscape. 7 of those were considered remote waterholes, so they did not have any sort of permanent human infrastructure or buildings. And then 5 of them did have buildings located at them. So just so I don't have to say permanent infrastructure over and over again, we call these lodges, but just know that the infrastructure means any sort of building or lodge that humans are using or accessing at some point. So the study was conducted during the annual 72-hour game counts that occur around September or October of every year. And we deployed motion-activated reconyx cameras for 72 hours before those counts, 72 hours during the counts, and 72 hours after the counts in order to capture the periods before, during, and after human presence. For our image analyses, we considered an event to be independent if it occurred more than 30 minutes from the last image of the same species, and we totaled the number of visits for each species and removed any species from analyses if they had fewer than 30 visits, just due to low sample size. Then we looked at the number of visits at waterholes, time spent at waterholes, and diel activity patterns around waterholes as a function of period. And period is always meaning the 72 hours before counts, 72 hours during counts, and 72 hours after counts. We also compared waterhole type. So again, this is the remote waterholes versus those that we're calling lodges. And then time of day, so just comparing day and night. For our analyses, we used generalized linear mixed models with Poisson distributions to look at the number of visits to waterholes. For time spent at waterholes, we used linear mixed models. And then for diel activity patterns, we calculated the coefficient of overlap values using kernel density estimates with 95% confidence intervals, and then compared activity patterns between each period using Mardia-Watson-Wheeler statistical tests. We also compared remote waterholes to lodge waterholes for diel activity patterns, and we found no differences between the two groups, and so we pulled all of the waterhole data together for diel activity patterns and didn't separate them out any further. We had a total of 17 species for our analyses after we removed those that were either too small of body size or too low of sample size. So 5 of these were carnivores, and then 12 of these were herbivores. With a total of 216 trap nights and about 4,500 images that we used for all of the analyses. So looking at our results, for this slide and the next, I have tables that look similar to this. So the minus sign indicates a negative effect in our model results, and the plus sign indicates positive, which I'll explain a little bit more. First, we looked at the number of visits to waterholes, and we didn't find any difference in the number of visits based on period or human presence. We did find that the black-faced impala, elephant, kudu, plain zebra, and springbok all visited the waterholes less often at night and more during the day. And the duiker visited more at night and less during the day. So those results were expected and not anything surprising. The black-faced impala did visit lodge waterholes more often than remote waterholes. For time spent at waterholes, we found that giraffe spent more time before the counts than they did during counts. And there were no significant differences between during and after counts. The spotted hyena spent less time before counts and more time during counts at the waterholes. Elephants, spotted hyenas, and springbok all spent less time at night at waterholes than they did during the day. So the meat of our results is really in our diel activity patterns, so I'll spend the most time talking about these. Just to give you an idea of what you're looking at with diel activity patterns, we visualize these using kernel density curves, so that these show a distribution of data over a continuous time period. So on the x-axis, we have time of day, including a full 24 hours from midnight to midnight, essentially. And then on the y-axis, we have density, or really the number of visits during that time of day. I have each period indicated with a specific color and line type. So before counts is in the peach color, during is blue, and after is green. At the top of each figure, these are our coefficient of overlap values that are calculated between 2 periods. So B stands for before, D stands for during, and A stands for after. We compared 2 periods together. A coefficient of overlap value ranges from 0 to 1, 0 meaning no overlap at all. So that would mean they've visited the waterholes completely different times. And then 1 is complete overlap. So that would mean they're visiting at the same time each day. And In the parentheses, we have 95% confidence intervals, and then the asterisk indicates that there is a significant difference between the activity patterns for those two periods. Out of our 17 species, 14 of these had differences in activity patterns between at least two of the periods. So first we'll look at during counts versus before counts. So this is during human presence compared to before human presence. We found these species were more active during the day, during counts while humans were present, relative to before counts. Okay, you can see this if you look at the kernel density curves with that big blue peak in the middle of the day. That's indicating more activity during those daytime hours in the middle. So we saw this for the lion, spotted hyena, giraffe, gemsbok, impala, and steenbok. The opposite pattern, these species were more active during the night, during counts relative to before, when they were more active during the day. So again, you can see that peach peak in the middle of the day here for the jackal, duiker, elephant, springbok, and mountain zebra. Now looking at during counts versus after counts. So during human presence relative to after they left. These species were more active during the day, during counts relative to after. So that's our lion again, brown hyena, jackal, giraffe, and steenbok. And then these species had opposite patterns. So they were more active during the night while humans were present and more active during the day after they left. That's the duiker, the plain and mountain zebras, and the springbok. And now looking at before counts versus after. So before human presence compared to after human presence, these species were more active during the day before humans came, and then became more active at night after they left. That's the leopard, brown hyena, jackal, steenbok, and elephant. And these species were more active at night during before counts versus after. That's the impala, plain zebra, giraffe, and gemsbok. So that's a lot of results, and just to kind of sum up the big takeaways from all of those, we did see that during counts, these 4 carnivores that are typically nocturnal or crepuscular became more active during the day while humans were present, relative to before and or after the counts. So that's the brown hyena, jackal, lion, and spotted hyena. And these 4 ungulates became more active during the night, during counts while humans were present, relative to before and or after the springbok, duiker, mountain zebra, and plain zebra. So what we found based on our results is that because there were no differences in the number of visits to waterholes based on period, and because for most of the species time spent wasn't really affected, we don't think that human presence for game counts in order to estimate population sizes is going to bias any of the results, especially in somewhere like a game reserve where the animals may be more habituated. We did find, though, that 14 species altered their diel activity patterns around human presence to some extent. We found that because spotted hyenas spent more time at the waterholes while humans were present, this could have increased it could mean that they increased their vigilance while humans were present. If anybody's sat and watched an animal at the waterhole, you can kind of understand why this might happen. When they're approaching the waterhole and looking around, they're spending a lot of time trying to assess their risk for approaching the waterhole and letting their head down to drink. They spend a lot more time than if they were not scared and simply approached the waterhole and started drinking, kind of like you see in this video here with the gemsbok. Most notably, we found that those 4 crepuscular or nocturnal carnivore species became more diurnal when humans were present, and we believe that in response to their diurnal activity, those 4 ungulate species became more nocturnal in order to avoid predation. So as such, it's important to note that modifications of temporal activity patterns due to human presence could lead to altered predator-prey interactions, which can lead to altered ecosystem processes. It can also disrupt circadian rhythms and affect reproductive fitness, which can impact a species' overall health. As humans continue to kind of encroach into wildlife habitat and wildlife-based tourism increases, it's important that we monitor any behavioral changes in wildlife populations in order to ensure that our conservation efforts are progressing while also managing the tourism industry and making sure that it's beneficial as well. And with that, I'll take any questions.